Good morning, all students. And today we will be continuing with the discussion of the chapter Respiration in Plants. And today we will be discussing the anaerobic respiration, that is a fermentation. We will be proceeding with this. So first of all, before going into the discussion of anaerobic respiration fermentation, I would like to highlight over here that why we are moving to the discussion of this part, because already let me come to that slide. Yes, because when we were discussing the glycolysis, there we said that the glucose converted into pyruvate, and the pyruvate is then converted into acetyl-CoA, which then enter into the TCA cycle of the mitochondria for the generation of the ATP. This is the case when the oxygen is present. But what will happen when the oxygen will not be present? Though the pyruvate conversion into acetyl-CoA and then the acetyl-CoA entry into the citric acid cycle will be also our part of discussion. But before that, we need to discuss the pathway. As we said that pyruvate is a replication fork. Now this pyruvate enters into the anaerobic pathway. This part is the anaerobic pathway, which I'm writing with A. So this pathway is the anaerobic pathway. This part we have to discuss. Okay. So let us discuss this part. Yes. Now see. Fermentation or the anaerobic respiration. If we are defining, if we want to define the fermentation, then definitely we will be saying that it is the incomplete oxidation of glucose under anaerobic condition. Definitely it means that when it is taking place in absence of oxygen. So where it occurs, in which type of cell? In many prokaryotes and unicellular eukaryotes. And if we are discussing about the types of fermentation, there are two types. One leading to the formation of alcohol, which in the pyruvate giving as or serving as a fork in the process of the respiration. There also we have seen. One is alcoholic fermentation, which leads to the formation of alcohol. And the another is the lactic acid fermentation, which lead to the formation of the lactic acid. Both are we can say in an anaerobic condition, one taking place in complete absence of oxygen and one taking place in inadequate supply of oxygen. But both the condition, we will be classifying them as anaerobic. So fermentation is definitely the incomplete oxidation of glucose under the anaerobic condition, and it is of two types: alcoholic fermentation and lactic acid fermentation. Next. First, we will be discussing the pathway where the pyruvate or the pyruvic acid produced as a result of the glycolysis undergoes alcoholic fermentation or the anaerobic pathway. Here, see what happens. We know that, here, we know this part, glucose being converted into ultimately the pyruvic acid. This we are knowing from the glycolysis, this part. This part is your glycolysis. Now this pyruvic acid, when we can say is converted into ethanol and CO2, there NADH is being converted into NAD+. Okay, first we'll be discussing the pyruvic acid conversion into the ethanol. This part we will be discussing. So in case of the alcoholic fermentation, the pyruvic acid formed from glucose is converted into CO2 and ethanol. the enzyme definitely which catalyzes this reaction are the pyruvic acid decarboxylase and alcohol dehydrogenase. And for example, if you are talking that where in which organism this process is occurring, definitely it is the example where in the yeast it is occurring. And the yeast poison themselves to death when the concentration of the alcohol reaches about 13%. If we are seeing in a brief or we can say in a one of the diagrammatic way, the sugar is being consumed by the yeast where it is, or we can say we will be saying consumed as because it is a case of the anaerobic respiration, and there it is forming ethanol, energy and CO2. But the amount of the energy definitely will be less here, as because it is a case of the anaerobic respiration. So the pyruvic acid is converted into ethanol over here, and the net, yes, the net ATP production from fermentation of one glucose molecule is 2. That means 2 ATP is being produced. Okay. So the net gain from the fermentation of one glucose molecule is the 2 ATP. 4 ATP was from the glycolysis minus the 2 ATP utilized. So ultimately it will be your 2 ATP. Is it clear to all this portion? Now this part is clear to all? Yes, through the chat box, you just give me the reply. Okay, fine. Okay, yes. Yes, all the students. Okay, fine. Now, let us move to the next part. Next, we will be discussing about the anaerobic respiration, the pathway that is lactic acid fermentation. Here again, you can see this part, the pyruvic acid being converted into the lactic acid. So here pyruvic acid is converted into lactic acid. This is the case with some bacteria. In animals, when oxygen is inadequate, remember student, this is very important part. Though we classify this also as anaerobic condition, but the basic difference between the alcoholic fermentation and the lactic acid fermentation, definitely with their end product. But one more thing is there, that is the whether oxygen is available in ample quantity, or it is inadequate, or it is completely absent. So in case of the lactic acid fermentation, the oxygen is inadequate during exercise. In our muscles, we feel pain. That is due to the accumulation of the lactic acid. So the pyruvic acid in the muscle cell is reduced to lactic acid by lactate dehydrogenase. So the pyruvic acid in the muscle cell is reduced to lactic acid by lactate dehydrogenase. And in this process, we are seeing that the pyruvic acid is converted into the lactic acid. Is it clear to all? Is it clear to all? Okay, fine. Okay. Okay, Pragya. Okay. I said that in case when we are doing vigorous physical exercise or we are running for a longer period of time, then we feel pain in our muscles. That is called the muscular cramps, and which is due to this lactic acid formation. But this process does not take place in absence of oxygen. Oxygen is present, but the amount is not sufficient to completely break down it, to give the main product of the respiration, that is CO2, H2O and energy. Rather, here lactic acid is being formed, and this lactic acid is, or we can say the enzyme which catalyzes this lactic acid formation is lactate dehydrogenase. Is it clear to you, Pragya, now? Okay, fine. Okay. Okay. Okay, fine. Now, the next part. Now, okay. Now, there is some drawbacks also of the fermentation. The first most important drawback is the energy production, as because we know that this is an anaerobic condition where either incomplete oxidation or incomplete breakdown of the respiratory substrate takes place. So the definitely the energy production is also limited. Here, less energy, or we can say less than 7% of the energy in the glucose is released, and not all of it. It is trapped as a heat energy bond of the ATP. And definitely, if we are talking about the products, acid or the alcohol, they are definitely the hazardous product. Can you say me, student, why this the acids or the alcohol are named as hazardous product? Who want to answer? I will unmute them. Just raise your hand. Why this acid or the alcohol are the hazardous products? Why we are classifying them? So these are definitely the drawbacks of fermentation because of the products which are being formed. Can anyone say why we are classifying the acid or the alcohol as a hazardous product? Anyone? Okay, Amrita, you can unmute yourself to answer this question. Yes, Amrita.
Sir, I am not able to.
You won't be able to answer?
Sir, I am not able to answer this question.
Okay.
No, sir.
Okay, okay, no problem. Let me then, okay, Prashant. Prashant, you can unmute yourself.
Yes, sir. No, sir, I can't answer.
You also can't answer. Why? Why the acid and the alcohol are the hazardous product? These are the awareness-based topic. Okay. Pragya.
So, because I think acid and alcohol, their accumulation in the cells cause disruption in the cellular activities, and they are poisonous though. Like they cannot be oxidized further to generate ATP,
Yes,
and they
definitely.
are not very useful for the cells also.
Yes, and though one very important thing also that we are using them for creation or for making up of the different varieties. And definitely, if you are talking about the alcohols or if you are talking about the acids, they are definitely from we can say the metabolic pathway point of view or the health point of view, they are definitely harmful. So that is why we are saying that the acids or the alcohol are the hazardous product, and this is the one of the very important drawback of fermentation. Now, is it clear to all the students?
So
Is
I have read it.
okay, Pragya,
So I
yes,
have
go
read
ahead.
it in the book that the lactic acid, which is like accumulated in our cells, which causes cramps, like in the when we are doing exercise or are in some vigorous physical activity or are involved in some physical activity, then
Correct.
this lactic acid is again transported to our liver, in which where it is broken down to by a process gluconeogenesis. Like, am
Gluconeogenesis,
I
yes.
saying it right?
Yes, yes, correct,
So, sir,
Pragya.
it is then again converted into glucose, and
Yes.
then it is broken down in
Yes,
us.
yes, then it is broken down. Correct. Okay, Amrita.
So how can we see that? Yeah, truly what damaging or hazardous when lactic acid is again mm, converted into glucose in our liver by the process of gluconeogenesis, which does not require oxygen.
Okay, okay, Pragya. Okay, I'm coming to your question. Yes, just yes, I think now you can mute yourself, Pragya. Okay, and meanwhile, Amrita has also okay, okay. See, yes, okay, Amrita. Yes, Amrita also said to repeat once again. Definitely, students, as we are knowing that when these lactic acids are being formed, definitely the cramps occur. So this part becomes, or we can say this is contributed toward the disadvantage. That is the formation of the lactic acid. And if we are saying about the alcoholic fermentation, which is occurring in the yeast, you must be knowing the application of the alcoholic, or we can say the alcohols. So definitely the wines, the beverages, they are not the advantageous product if consumed in excess quantity. And that is why they are regarded to be as a hazardous product. Okay, is it clear to you, Amrita, and all the students? Okay. Okay, fine. Okay. Yes, I got the feedback, all the students. Okay. Now let me come to the next part. Now we will be coming to the aerobic respiration. Why we are again shifting back toward this part? Because ultimately till now we have not completed the mechanism of aerobic respiration, though the universal step of respiration we have done. Student, can you tell me which what is the universal step of respiration? Immediately through the chat box you replied to me. What is the universal step of respiration? Very good, Prashant, very quick. Yes, correct. This answer I want from all the students. What is the universal step of respiration? Okay, very good. Glycolysis, correct. So we did with glycolysis, and now when the glucose is converted into pyruvic acid, this pyruvic acid will now further be converted into acetyl-CoA, which will enter into tricarboxylic acid cycle or the Krebs cycle, thus starting the second step of the mechanism of respiration. So let us move now. Before going into the discussion of the TCA cycle, a brief discussion regarding the aerobic respiration. Now, the aerobic respiration is definitely a complete oxidation of organic substances in the presence of oxygen, releasing carbon dioxide, water, and energy. This we know from the very well definition of the aerobic respiration. And definitely if we talk about the cellular organelle where it takes place in the mitochondria. And for this, the pyruvate, which is a final product of glycolysis, is transported from the cytoplasm into the mitochondria. Student, if you will have a look over this picture, which is showing you the summary of the aerobic respiration, then definitely here you can see in this image the glycolysis in which glucose is converted into pyruvic acid, and here ultimately 2 ATP molecules are being produced or given out also. This pyruvic acid now enters into the Krebs cycle. In the Krebs cycle, CO2 is being produced, and here now the chemical energy, as we know, through the electrons. So these electrons are transported through the ETS or the electron transport system, and ultimately in this process, the net gain of ATP is 38, even 36 we say, 38 molecules of the ATP, and water is also released as a byproduct during this process. So this very diagram show you the overall summary of how the different step of the respiration occurs. And then part wise we will be also doing this. So now let us move to the next part. Yes. If we talk about the introductory part, crucial events of the aerobic respiration, complete oxidation of pyruvate by stepwise removal of all the hydrogen atoms, leaving 3 CO2. It takes place in the matrix of the mitochondria. Passing on of electron removed as part of hydrogen atom to molecular O2 with simultaneous synthesis of ATP. It occurs in the inner mitochondrial membrane. Definitely, student, this is also a very important part, that complete oxidation of the pyruvate is occurring in the matrix of the mitochondria. But passing of the electron, I told you, which is the electron transport system, occur on the inner side of the mitochondria. Okay. Next. Now see how the pyruvic acid at the initial step, the pyruvate or the pyruvic acid enters mitochondrial matrix and undergo oxidative decarboxylation in presence of the enzyme pyruvate dehydrogenase. And it requires a coenzyme such as NAD+ and coenzyme A. Student, here I would like to ask any of the student, do you know the meaning of the word coenzyme? As because in biomolecules also we have discussed that what do you mean by the term coenzyme? Coenzyme we have discussed in biomolecule. Quickly raise your hand who knows the answer. What do you mean by the coenzyme? So that I can unmute you. Let me see the participation list also. What do you mean by coenzyme? Yes, for this, I'm giving, I'm unmuting Anshu. Anshu. Yes, okay. Okay. Okay, Pragya, you want to answer? Acha, okay. Pragya, you have sent one question also. Okay, I will come to that. Okay, yes, Khushbu also. Okay, let me this time give chance to Khushbu because Pragya, yes, always answers. Okay, Khushbu, just a moment. Yes, Khushbu, you can unmute yourself. You can unmute yourself, Khushbu.
Yes, sir.
Yes, Khushbu, what is a coenzyme?
Sir, coenzyme is an organic non-protein compound that binds with an enzyme to catalyze the reaction.
Okay, very good. Definitely a non-protein compound that bind with the enzyme to catalyze. That means it increases the rate. Definitely, Amrita said organic compound which is associated with apoenzyme, correct? Very good. Okay. Yes, Pragya. Yes, your question. Yes, definitely. It is not truly hazardous, as you are correct, because again, it is being converted into the glucose through the process of the gluconeogenesis. But due to the very first step, or we can say the beginning step of the muscular cramp formation, it is said to be hazardous. Okay, Pragya? Is your doubt clear? Yes, Mihir, correct. Organic molecule that bind to the active site of the certain enzymes. Correct. Okay, fine. So correct. So the pyruvate, the pyruvic dehydrogenase require the coenzymes such as NAD+ and the coenzyme A. And during this process, 2 NADH molecules are produced from 2 molecules of pyruvic acid. And the acetyl-CoA then enters tricarboxylic acid cycle. That means the acetyl-CoA will now enters into the TCA cycle. So here you can very well see that how the pyruvic acid plus the coenzyme along with NAD is converted into acetyl-CoA, which is we can say the molecule that will enter into the TCA cycle or the tricarboxylic acid cycle. Now, now we are coming to the steps of the tricarboxylic acid cycle. This cycle definitely due to its discoverer Hans Krebs, it is called the Krebs cycle, and because of the formation of tricarboxylic acid, it is called the TCA cycle, and this is also called citric acid cycle because of the first stable product. Now, succinyl-CoA is converted into succinic acid. 1 minute. Yes. See, first of all, an overall view of the different steps of the TCA cycle. Here, the 5th, 6th, and 7th point has been indicated, and the rest we will be discussing from the schematic diagram only. We all know pyruvate is being converted into acetyl-CoA. Now, here the coenzyme is given out back, and now the first product, that is a citric acid, is being formed. This citric acid, during this process, or we can say the citric acid then form alpha ketoglutaric acid with the liberation of the CO2 and the conversion of NAD into NADH. This alpha ketoglutaric acid with the utilization of GDP and NAD plus, and here again also the evolution of CO2 takes place. This is being converted into succinic acid, and this succinic acid is converted into the malic acid with the utilization of FAD. The malic acid then gives back the 4-carbon compound, that is oxaloacetic acid, with the utilization of MAD. So here we are seeing, if we want to summarize what is happening, the crucial events which occur in the TCA cycle are, first of all, at 3 points of the TCA cycle, NAD plus is reduced to NADH. This is the very important part, because many times it is being asked that at what places this process is occurring. So at 3 places, when the citric acid is converted into alpha ketoglutaric acid, and alpha ketoglutaric acid is converted into the succinic acid. Here 2 times, or we can say NAD is reduced to NADH, and also when malic acid is converted into oxaloacetic acid, there also NAD is converted into NADH. Now at 1 point only FAD is reduced to FADH2, and that is definitely during the conversion of the succinic acid into the malic acid. Continued oxidation of acetyl-CoA via the TCA cycle require continued replenishment of OAA, because the cycle has to run continuously, and it also requires regeneration of NAD and FAD from NADH2 and FADH2. So this is the overall summary of the equation involved in the Krebs cycle, where you can see what are the reactants and what are the product. Pyruvic acid, 4 NAD plus 2 molecules of H2O, 1 molecule of ADP and inorganic phosphate in the mitochondrial matrix, given the product 3 molecules of CO2, 4 NADH, 4H plus, FADH2 and ATP. Thus, glucose is broken down to give 6 CO2, 8 NADH, H plus 2 FADH2 and 2 ATP. Student, can you say that this equation, as we have said, like this one, how it is being converted into the double quantity over here? We said that the products are 3 CO2, 4 NADH, 4 FH plus, FADH2 and ATP. How it is being converted into double? 6 molecules of CO2, then 8 molecules of NADH, 8 molecules of H plus, then 2 molecules of FADH2 and 2 molecules of ATP. How it is being converted? Who wants to give the answer? Raise your hand quickly. How we are saying that ultimately it is converted into double the quantity? Okay, just let me see the chat box. Okay, okay, Pragya. Yes, I've seen your query. Okay, Aditi, also I've seen your query. Okay, Aditi, I'm coming to your question also. Aditi, coenzymes are definitely the organic molecule that bind, or we can say it is an organic compound which is associated with the apoenzyme. That means the protein part of the enzyme. The non-protein part of the enzyme which is associated with the protein part of the enzyme. Okay, sequentially I'm coming to your queries. Aditi, is it clear? Your confusion is clear? And then I will come to Pragya's question. And then I have seen Prashant has answered the question which I have said. Okay, Aditi. Yes, Prashant has given the correct explanation that as 2 molecules of the pyruvic acid is formed during glycolysis, for 1 molecule of the pyruvic acid, we are saying 3 CO2, 4 NADH, 4 H plus, and 1 FADH2 and 1 ATP is produced. So for 2 molecules of the pyruvic acid, the quantity will be doubled. This part is clear to all. Then I'm coming to the Pragya's query. This part is clear to all. This part is clear to all, that how the quantity is doubling. Okay, fine. Okay, okay. Once again, I re-explain the mechanism. Once again, I'm just a moment. Yes, see here in this mechanism, pyruvate, which was formed after the glycolysis, combines with acetyl-CoA to form the citric acid, which is the first product. This citric acid then is being converted into alpha-ketogluteric acid by the conversion of NAD into NADH. The alpha-ketogluteric acid is converted into succinic acid. Here, GTP is being utilized and NADH is converted into NADH. NAD is converted into NADH. The succinic acid is then converted with the utilization of 1 molecule of FAD into the malic acid, and the malic acid, for the cycle to run continuously, it regenerates oxaloacetic acid with the utilization of the NAD. So ultimately here 4 NAD is being required, 2 H2O is required, and 1 ADP is required, which gives ultimately the CO2, 4 NADH, 4 H plus, and FADH2 and ATP. Okay. Okay, Pragya, is it clear to you now? As you said, to once again go for the rediscussion of the cycle. Pragya, is it clear to you now? Okay, fine. Okay. Yes, I have seen. Okay, in the chat box now, I am seeing no more queries regarding the. Okay, students. So all of you, definitely today, your assignment will be that to practice the TCA cycle. Okay, you will be practicing the TCA cycle today, the semantic diagrams. All of you write down today's assignment. Practice the semantic representation of the TCA cycle. And the electron transport system will be our discussion of the next class. Practice the semantic diagram of the TCA cycle. Okay.